Hi, everybody, and welcome to today's program, the latest genetic and archaeological evidence for the peopling of the Americas, which is part of our ongoing Hot Topic series. My name is Brianna Pobener, and I'm a paleoanthropologist and educator at the Smithsonian's National Museum of Natural History. I'm a brown and gray-haired woman in a green and black jacket in front of a Zoom screen with an African savanna photo with grass and an acacia tree behind me. Whether this is your first time joining us or you've attended before, we're so glad to have you here. Before we get started, I have a few housekeeping notes. This discussion offers closed captioning. You can turn on the captions or off via the CC button, which should be located at the bottom of the Zoom interface. We're in a webinar format, so we can't see or hear you. If you have questions, please go ahead and submit them to the Q&A box, which is at the top or bottom of your screen. It has two speech bubbles, so we can sort through as many questions as possible. And the Q&A time really flies by. The Q&A box is also where we will share any relevant links during the program, so keep an eye out there. We'll start with an opening presentation by our speaker, Dr. Jennifer Raff, and then I'll join her here to take your questions. During her presentation, I'll also write answers to some of your questions, at least any that I can answer, as will another member of our behind the scenes team, GWU, George Washington University, PhD candidate Ryan McRae. Now I'd like to go ahead and introduce our speaker. Dr. Jennifer Raff is an associate professor of anthropology at the University of Kansas with a dual degree PhD in both anthropology and genetics. She specializes in anthropological genetics related to the initial peopling of the Americas and subsequent histories of indigenous populations throughout North America by studying ancient and contemporary genomes. In addition to her research, she's been writing on issues of scientific literacy, genetics, and anthropological research at her own website, Violent Metaphors, and for The Guardian, Forbes, HuffPost, and Evolution Institute blogs for several years. She is also the author of the New York Times bestselling book, Origin, A Genetic History of the Americas, published in February 22. Thank you so much, Dr. Raff, and we're looking forward to your presentation.
Thank you so much. I'm so honored to be here. I really appreciate it. And hello to everybody who's joining us. I'm going to start the presentation just with a little bit of history. So I'm actually speaking to you right now. Oh, sorry, I should start by saying that I am a dark-haired woman wearing a green shirt and a black jacket, sitting in front of a bookshelf with a variety of books and other things on it, including my own book and a poinsettia. So I'm speaking to you right now from the University of Kansas in Lawrence. And the land that KU occupies was taken from the Kaw or Kanza, Osage, and Shawnee nations. Kansas, the lands that are called the present-day state of Kansas, have a very complicated history of settler colonialism. So tribes have been forced into and through and out of this area before it became a state. But today, the state of Kansas is home to the Prairie Band Potawatomi Nation, the Kickapoo tribe in Kansas, and the Sac and Fox Nation of Missouri in Kansas and Nebraska, and the Iowa tribe of Kansas and Nebraska. In addition, because Lawrence is the location of Haskell Indian Nations University, many American Indian and Alaskan Native peoples from across the region have ongoing ties to this place. So understanding and acknowledging the history of places throughout the Americas is an important step in equity and justice for past harms, but it's really only a first step. Members of our research group try to go beyond just giving land acknowledgements and return tangible benefits to Indigenous communities with whom we work and on whose lands we work and live. And if you're interested in how we do that, I'd be happy to chat with you about the specifics during the Q&A. So today I'm going to be talking to you about the latest scientific understandings of how the first peoples came to the Americas. These were the last continents to be explored by modern humans and the last steps on our global journey. And we still have many questions about this process. We once thought archaeologists and geneticists once thought we had a really good idea of how this happened, but we've had to revise our models a lot in the last few decades. And in no small part, this is due to the introduction of a new kind of data, complete genomes from ancient and living indigenous peoples. We're learning so many new details each year that it's really hard even for specialists to keep up. But today, what I'm hoping to give you is an accessible overview of the state of the science, and also a discussion about some of the ethics and complications that are involved in doing this work. And I hope that when you hear of new results and new studies that come out in the future, that you'll have a framework from which you can understand these new discoveries. But as we go through this talk, please be aware that there have been many nefarious attempts to use stories about Native American origins for disenfranchisement, land theft, and even genocide. And we need to be mindful of this history, even as we're learning about the latest scientific discoveries. We need to keep in mind how those discoveries have been made in the past and how they are being made today. It's not something I can really do justice to in this short talk, but I'm very happy to talk about it more in the Q&A afterwards. And I do cover it in my book as well. Okay, I also want to start by acknowledging the Indigenous and non-Indigenous scholars and organizations whose work has influenced my thinking on these issues. A lot of what I'm going to be talking about in this presentation is built on a foundation of Indigenous and academic and public scholarship. And these are some, but by no means all, of the people whose work has influenced my approaches to research. In addition, the research that I'm going to be presenting here and describe in my book is the work of many, many geneticists and archaeologists. And it's a career-long goal for me to try to help make the scientific literature accessible to people who are not specialists. And so in order to help you delve deeper into some of the topics that I'm going to be presenting today, I have created a readings list and a Google Drive folder in which I will be dropping PDFs of the papers that I'm talking about today. And I couldn't finish it all before the talk. Unfortunately, I ran out of time, but I will be doing this all day long. So if you get the link, click on it, you'll be able to access it, and you can access the research papers directly through that. So this is going to be kind of changing all day, but so I hope that this is going to help break down some of the barriers between academic scientists and the public. And if you have other ideas for how we can do this, I would love to hear from you. I'll give you some ways to reach me on the last slide. Okay, so I want to start our exploration in a way that a couple of my archaeological colleagues have done in their talks, which I really like. And this is a bit of archaeological history that starts with a really fascinating and paradigm-changing important person in history. So we're going to go back to the year 1908 and the Crowfoot Ranch near Folsom, New Mexico. So it's September and the air is hot and the cowboys are riding through the Wild Horse Arroyo looking for stray cattle after a major flood that nearly wiped out the town of Folsom. So this man, George McJunkin, was riding, he was the foreman, and his attention was caught by a pile of bones at the base of the arroyo. And he recognized at once that these were not the remains of one of the cattle that had drowned, but instead these were old and dry. McJunkin knew about animal bones from his experience working with horses and cattle and hunting bison. So he inspected the remains and determined that they were not of a cow, but of a bison. And in fact, a bison much larger than any living creature he'd ever seen. So he removed some of the bones from the site and tried to get people interested in it, which unfortunately, the site was not excavated until after his death. But McJunkin his find was paradigm changing in the history of archaeology. His recognition of the importance of these remains was a reflection of his experience as a cowboy, and also as a well-educated and self-taught naturalist. So George McJunkin had been born into slavery in Texas and had been living as a free man since the end of the Civil War. He left home to take up the life of a cowboy and traded horse breaking lessons for reading lessons. Once he began to read, he read everything he could get his hands on and became a very experienced naturalist, which meant that he was positioned to be the right person in the right place at the right time to recognize the significance of his discovery. These partially mineralized bones turned out to have been the remains of an extinct animal from the Pleistocene. And this became known as the Folsom Site, and it would eventually reveal to archaeologists dozens of extinct bison remains, many associated with spear points like the one seen in this photo. This helped establish that humans had been present in the Americas since the Pleistocene, rather than being latecomers within the last 5,000 years, as many scholars believed. So by searching for extinct animal remains, archaeologists began to find more evidence of the humans that hunted them. And following the discovery of Pleistocene age artifacts at sites like Folsom, archaeologists began to identify many more early sites. And as radiocarbon dating made it possible to establish absolute chronologies beginning sometime in the 1950s, a new model began to take shape that explained how people first got to the Americas. And this has come to be known as the Clovis Model. If you're my age or older, this is probably the model you learned in school. So this took into account the apparently sudden and widespread appearance of people who made the Clovis technologies, which are characterized by bifacially fluted spear points like this one. And they appear in the archaeological record of North America around 13,000 years ago. And as this map shows, they are widely distributed. These sites were taken as evidence of the first peoples in the Americas. And in this model, this Clovis model, the ancestors of the first peoples had come from Siberia, where they led a rugged existence hunting these massive ice age beasts. They lived in bands, small bands of extended families, and they ranged widely over their lands following their prey. These ancient Siberians crossed the Bering Land Bridge near the end of the late Pleistocene. And the Bering Land Bridge existed between about 50,000 and 11,000 years ago during a period called the last glacial maximum, which is a period of global cooling and dry conditions. And it reached its peak between about 26,000 and 19,000 years ago. This played a key role in understanding the peopling of the Americas. So during the LGM, enormous quantities of water were bound up in glacial ice masses that covered enormous stretches of continents, including most of North America. And so the coastlines were very different than they are today. So ocean levels were approximately 100 meters lower on average than they are today. And this caused this enormous land connection to be created between Asia and North America. And this is a GIF of coastline changes over time here, beginning about 21,000 years ago. So the LGM was a really harsh time in which to live. So it was bitterly cold, so dry that large regions of the Northern Hemisphere were actually deserts. And so this affected the distribution of plants, which affected the distribution of animals, which of course affected where humans were able to live. And so around the world, the archaeological record shows people retreating into climate refugia during this period. So the conditions of the LGM also prevented people from moving very far into the Americas. So they could get into Alaska, but they could not move out of Alaska into the rest of the continents because the way was blocked by this massive ice sheet formed from the joining of the Laurentide and Cordilleran glaciers. So potential routes of entry only began opening up as the Earth slowly warmed while the LGM was ending. And the first route along the Alaska coast opened up between about 18,000 and 17,000 years ago. The second route was in the interior of North America. And estimates for when this route first became accessible are kind of varying, but it was likely open and had animals moving through it by about 13,200 years ago. So according to the Clovis model, Siberian big game hunters crossed the Bering Land Bridge rather late. They traveled down the ice-free corridor to populate North and eventually South America just about 13,000 years ago. So this was an elegant model that most archaeologists believe accounted for nearly all of the archaeological and environmental evidence, and it was so strong that it persisted for nearly 50 years. But as it happens in science, evidence kept appearing that didn't quite work with this model. So better dating methods gave us a more precise estimate of when Clovis first appeared, but evidence kept popping up that predated Clovis. And eventually, evidence from the site of Monteverde in Chile here was broadly accepted as showing that people were present in the Americas by at least 14,200 years ago. So this was a major paradigm shift, and it raised all kinds of new questions. So archaeologists began to rethink how the peopling of the Americas might have occurred and began looking for other sites that might predate Clovis. These are just a few of them. There are so many other candidates for pre-Clovis sites. I just wanted to show you some of them. But the age of these sites made the ice-free corridor route impossible because they predate the opening of the corridor, at least our current estimates of that, and some by thousands of years. So, and in addition, the earliest evidence of people, archaeological evidence of people within the ice-free corridor, are actually people moving northward rather than moving southward. So the alternative route, the coastal migration by boat, would have actually made it possible for people to arrive at these different sites in time. And so I'm going to lay my cards on the table here and say that I find the Pacific coastal migration route to be more persuasive and a better explanation of the archaeological and genetic evidence. But I want to emphasize the fact that by no means do all archaeologists agree on this. In fact, there are some archaeologists who would disagree with any of these pre-Clovis sites. And we can talk about why that might be during the Q&A. Okay, but this is my talk, and I'm in the camp that interprets the totality of evidence as showing people in North America by at least 14,500 years ago, and probably a lot earlier than that. So that's going to be our working assumption for the rest of this talk. So this pre-Clovis presence raises all kinds of new questions. And these are summarized by my colleague here at the University of Kansas, Dr. Ted Gable, in a talk he gave a few years back. And I think he had this nicely encapsulates the research that the directions that research is going right now. As genetics has been increasingly applied to address these questions, it's given us a really complex set of answers. And we're going to tackle what ancient DNA has told us about these questions in the rest of the talk. But I want to start, before I start delving into the ancient DNA, I kind of want to caveat this. So there are many approaches to studying the past, and none of these approaches gives us a complete answer by itself. For example, we have these genetic stories, which I'm going to tell you, but in addition to that, many Native American societies have histories of their own origins, which have been transmitted mostly through oral traditions for many, many generations. Some of these histories align with the stories that scientific approaches tell, and some of them do not. And my focus is on genetics, but I want to respectfully acknowledge the many ancient perspectives on histories. Some of them are understood to be metaphorical. Some of them are taken to be literal explanations of origins. Events in the past unfolded in particular ways, but our ability to understand what happened is limited by the tools we use and the data that we have available to us. Far from being perfectly concordant, for example, archaeology and genetics sometimes seem to tell us different stories about the past. So just to be clear, I prioritize genetics as a tool for understanding the past in my own research, but I don't claim that the stories DNA tell are a complete answer. And they are not the only way to understand how history has unfolded. And the same is true for all of these approaches. So we have learned a ton about the biological origins of the first peoples through ancient genomes. And I'm going to spend the rest of the talk discussing this, but I wanted to briefly go into ways in which paleogenomics research is not like other genomics research first. So there's a couple of issues with ancient DNA recovery. So first, the process of recovering DNA from ancestors is almost always involves the disturbance of ancestors' remains. The DNA extraction process usually results in the destruction of a very small amount of bone or tooth or hair. And this non-destructive methods exist, but they're not very reliable. And so this fact alone takes DNA research off the table for a number of tribes and communities, but not all. but not all. So this is very much not a simple matter. And DNA is degraded and fragmented and extremely sensitive to contamination from the DNA of living people, which is abundant and intact. And aside from some exceptional cases, the majority of ancient samples have very poor preservation, and it's hard to get DNA from them. It's also technically challenging to take the precautions necessary to ensure that DNA from an ancient sample is uncontaminated. This is an image of one of our graduate students working in our lab, and it gives you an idea of the precautions that have to be taken. But when you do get DNA, an ancient genome, the histories it can tell are extraordinary. It's an archive of population genetics. The evolutionary forces that act on all populations, natural selection, genetic drift, mutation, gene flow, change the genetic composition of populations, and this evolutionary history can be reconstructed using the tools of population genetics. And ancient DNA is especially valuable because it gives us a direct window into past populations. So it can tell us at this time, we know that these genetic variants were present in this person at this geographic location. As of last year, these are the number of individuals from the Americas whose genomes have been sequenced. And this figure shows the number of complete genomes obtained using two different methods, which I'm not going to get into, with the size of the circle proportional to the number of ancestors whose genomes have been sequenced, and divided into 3 different time periods. So along with some key genomes from Asia and Siberia, these tell us a complex story. And I think of it as occurring in 3 major phases. So the formation of the ancestral population in Asia, the isolation of this ancestral population for several thousand years, and the dispersal of this population throughout the Americas. So I'm going to take you chronologically through the biological events that occurred, as far as we know, with existing data right now, in the process of peopling the Americas. And we're going to start with in the Upper Paleolithic in Asia. So first, all of the complete genomes recovered from the Americas so far show us that the first peoples have 2 major ancestry components. The majority of their ancestry comes from ancient peoples in East Asia. The other major ancestral component was discovered through the sequencing of the remains of a little toddler from this Upper Paleolithic site known as Malta. Archaeologist Kelly Graff, who is my colleague here at KU, was instrumental in arranging for the genetic study of this child, who was 3 to 4 years old and buried in a grave covered with red ochre. It was very ceremonial. And we know from his genome that he likely had dark skin and dark hair and brown eyes. And his population, which geneticists call the ancient North Siberians, was ancestral to both Native Americans and Western Eurasians and Europeans. So his population here experienced gene flow with ancient East Asians sometime around 23 to 20,000 years ago. And it was out of this meeting and mixing that emerged the ancestral population that gave rise to Native Americans. And it's marked as Beringian ancestors here. And also Paleo-Siberians who would then go back to repopulate Siberia after the LGM. We also see evidence for an ancestry component in some South American groups, which is related to present-day Australasians. And this is one of the really exciting areas of research in our field today. This is known as population Y. And I don't have it actually on this phylogeny here, but this ancestry component is really fascinating puzzle to geneticists. So we can actually rule out the most obvious explanation for it, which would be, a trans-Pacific migration by boat, because the genetic patterns seen in South America do not match the expectations for that model. And in addition, this ancient individual from Tianwan Cave in China also has this ancestry. So the most parsimonious explanation for these results is that both population Y and contemporary Australasians descend from a population that was widespread in East Asia. And the Tianwan individual would have been closely related to that. So we can talk more about the Q&A, about this in the Q&A if you like. I've got a couple of ideas, untested yet, but how they could have gotten into this population. So following the mixing of these different populations, the ancestors of the first peoples were isolated for several thousand years. And it's during this time that they evolved the genetic variants seen only in Native Americans. Many of us think that this pause must have taken place inside of Beringia itself, in one or more refugia along the southern margin of central Beringia, which would have had paleoclimatic reconstruction show us a more hospitable climate for people to live during this time period. But unfortunately, because this area is underwater today, we don't actually have any direct archaeological evidence of this or of this population anywhere. So this is an ongoing area of research in the field. As this population, ancestral to Native Americans and Paleo-Siberians, was isolated, it seems to have been splitting into several subgroups. So the ancient Beringians, who we know of from 2 genomes from these 2 sites in Alaska, they were a group of people who lived in Alaska at least until 9,000 years ago, although they don't seem to have any present-day genetic descendants. And another population that split off is we don't have any direct genomes from this population. It's called unsampled population A. We have only evidence of them from indirectly from their contribution to the present day Miche. So they the gene flow into the Miche's ancestors at some point in the past from this population. And the final group moved past the ice sheets and is genetically ancestral to Native Americans south of this region. So following the peak of the LGM, the Earth began to warm and glacial, as global temperatures rose, ice sheets began to recede and paths were available for traveling and populating new regions. And around the same time as when these paths began opening up, we see the emergence of at least 2 major groups of ancestors from whom all Native Americans south of the Arctic are descended. Northern Native Americans, abbreviated NNA, and Southern Native Americans, abbreviated SNA. These groups separate from each other sometime between about 17,500 and 14,600 years ago. There's a third branch that's very represented by genome from a single individual in British Columbia that splits off at the same time, not shown on this map. So we don't have a great understanding of the complete geographic distribution of SNA, but we know that everybody in South and Central and probably also in North America, south of Montana, which is where this site is from, they probably, all of these groups belong to the SNA branch. NNA, people descended from the NNA branch include Algonquians and Na-Dene, Salishan and Tsimshian speakers from Canada. And these different branches are likely diverged after these populations moved south of the ice sheets. And this gives us a clue that the movement southward happened very early and rapidly. So there's rapid genetic diversification over time in the SNA branch, suggesting that people were, as one of the authors of the study characterized it, leapfrogging across large portions of the intervening landscape. And so many of us interpret this as being consistent with the movement of people along the West Coast by boats, which would have allowed them to move much faster than land travel. And then, of course, at some point, we also have population Y getting into these populations. We don't know very much about that yet, so stay tuned. So this is kind of an excuse just to put cute pictures of dogs, puppies up. But in fact, we've actually been able to get some really important insights into human history from the genomes of dogs. So humans and dogs have co-evolved as domestic partners since the Pleistocene. And American dogs are shown genetically that they did not independently evolve from American wolves. They were actually brought to the Americas by the first peoples, and they are descended from Siberian dogs. And so this wonderful figure, which has been adapted from a study from Angela Perry and her colleagues, compares the branching patterns of dogs, and this is mitochondrial DNA, maternally inherited DNA. So here we have Northeast Asian dogs, and then these are the branches, the lineages that come to North America. And they've compared them to human lineages, and you can see how the branching patterns are very similar, as are the timings of these population splits. And so stay tuned for that. I think that we're going to learn a lot more about the history of the Americas from these amazing genomes, these amazing animals. So we have this neat story. The genetics seems to line up with the archaeology to argue for a early and rapid migration as soon as the coastal route becomes open, sometime after 18,000 years ago. Seems to work. But this neat story may be complicated by a recent, really exciting discovery in archaeology. So just as I was finishing up my book last year, a site was published that has evidence of human footprints. And these footprints have been dated, slightly controversially, to between 23 and 25,000 years ago, the height of the last glacial maximum, when we have no other evidence of people being in the Americas, except for some other sites, which I'm not going to get into, that are a bit controversial as well. So this is the White Sands Locality 2 site, located in the White Sands National Park in New Mexico. And it was once an ancient lake bed. The footprints have been dated by means of seeds of ditch grass that are embedded in them. And these footprints, these human footprints, we know they're from the Pleistocene because they're crossing over in the same strata as Pleistocene, extinct Pleistocene megafauna, these giant animals, mastodons and ground sloths and all kinds of wonderful creatures. And what's also wonderful about this site is that we can figure out the age of the people who left these footprints, and they see, but based on their height and stride length, and they seem to be mostly children and young people, which is really wonderful and gives you just an evocative image of the past. So I love this site. I will say that the dating has been questioned by a lot of archaeologists. The archaeologists who do this work, who've excavated the site, have pushed back on these claims. They have new dates we're waiting to hear about. I hope that we'll hear about them soon. This is the strongest candidate for a pre-LGM site that anyone has yet found. And I can't give you an honest assessment of whether this will ultimately be accepted by the archaeological community, but I sure am excited about it. And I am working hard to try to think about how to reconcile genetic models with this archaeological evidence. One factor which is complicating the genetic record is that we don't have many genomes from ancient peoples in North America, especially in the present-day United States. So you can see these large gaps here. There are many reasons for this. So I talked about the challenges of ancient DNA research, as well as the problem that we don't want to disturb Native American ancestors' remains if that is inconsistent with descendants' values. That's really, really important that we respect that. And there are other reasons. So, and a lot of these come down to the fact that indigenous peoples in the Americas have had their wishes violated in the pursuit of scientific knowledge so often. There's a very understandable lack of trust in researchers who do their work at the expense of, rather than in collaboration with tribes and communities. And so this is, there's a very important discussion about this going on in my field right now, how to do this work ethically, if at all. And I don't have time to do it justice here, but I do talk about it at length in my book, and I'd be happy to chat about this further in the Q&A if you would like to. And I will be dropping references to some key papers that discuss this into my Google Drive folder, so you can read them yourself. So the story that I've told you today, and I cover at length in my book, is not only of the far distant past, it's also the story of the present and more recent past. So how we have come to our understandings of the scientific origins, the origins of the first peoples, is just as important as what we think we know. And this history we have to understand is deeply rooted in colonialism, centuries of exploitation of indigenous peoples. And we also need to be aware that the results of our research today have the potential to deeply impact Native populations. And we can choose to do this research in a positive way. And I give a number of examples, positive collaborations with tribes and communities and non-Native scientists. But we can only do that if we have a good understanding of the ways in which previous work has harmed Native Americans. And so that's one of the major themes of my book. I try to interweave it with the science, and I hope that people can take from it an understanding of how complex and relevant this fascinating story is and why it matters. Okay, so these are ways to reach me. I am going to be difficult to reach for the next month or so, but I'm happy to try to answer any questions you might have. And thank you again for coming to this.
Fantastic. Thank you, Jennifer. That was a really wonderful, brief but informative overview of what we know about the peopling of the Americas. And we already have questions coming in, so I'm just going to jump right in. Here's an interesting question about methods that came in from Al, who says, please address the viability of using eDNA, which is environmental DNA from soil and sediments, alongside of aDNA, ancient DNA, from human remains in your or this type of research.
That is a great question. And I think that eDNA is the future of this field, or it's going to be one of the most exciting areas that we're going to see progress in this field in the future. I am not an eDNA expert. We actually have some in our research group who do this, but I think that what's really powerful about eDNA is you can reconstruct paleo environments from getting genomes. Now, that's present in the soils, in ancient soils. There's some caveats. So this field is very much in the early stages of developing its methods and developing standards of practice. And so I think that we're going to see an increase in rigor and also an increase in questioning results, because that's part of the natural scientific process, right? But there's been some really exciting studies that have come out recently using eDNA as a powerful tool to identify the presence of, for example, ancient hominins at a particular site, not relevant here, but in other places, and also reconstruct ancient environments. And I think that's really wonderful. And I would like to see more of this research. And I'm so excited about it.
So speaking of ancient hominins, we do have a question related to ancient hominins. Mark says, I have seen a speculation that population Y had introgressions from Denisovans. Thoughts on this?
Yeah, not just population Y, but indigenous, the first peoples we know have Denisovan and Neanderthal ancestry as well. And that does not seem to be any special introgression event outside of what normally took place among all ancient individuals in the Upper Paleolithic. So I don't think that, for example, Denisovans or Neanderthals were in the Americas, despite there's been one or two claims of archaeology that date to that time period. They're quite controversial, and I'm still not sold on that at this point. But yes, the ancestry of Denisovans and Neanderthals present in Native Americans can actually be attributed to those Upper Paleolithic introgression events.
So here's an interesting question from Dina, who first says, thank you for your presentation. That was actually not the question that I meant to ask now, but I can ask that now anyway. Do you think there is a use for a database for Indigenous biological information that is controlled by Indigenous groups, or THPO, to allow them to decide who gets to use that information?
Yes, that raises a really great point, which is Indigenous data sovereignty. And I want to give a shout out to some of my Indigenous friends, the Native Biodata Consortium, who are working to create databases and protections for Indigenous data that are maintained on tribal lands and protected, and so that the consultation process and the consent process can be done in a way that's separate from the normal processes that one has to undergo when you're subjected to, like say 3-year grant deadlines. I mean, it's a cycle this is a very academic issue, but it's really important, right? So we need to have ongoing relationships. Those of us who do research in this field, we need to have ongoing relationships with tribal communities with whom we work that outlast the normal span of a study, and have ongoing consent, because communities can change their minds about whether or not their data should be used in particular ways. And I think that's really important to honor. And it can be difficult for researchers who are tied to these funding cycles to maintain that, even if you have good intentions. And so there is definitely a great movement out there right now to enable the tools for tribes to do this. And I'm so supportive of that.
Fantastic. And I just got a notification that my internet is unstable. So if I freeze or at any point, just let me know.
Okay.
So here's a question from someone who with first initial L. Is it at all possible to use current DNA from populations to assist with this effort, or are the needed samples simply too far back in time?
Yes, great question. And when I have a longer talk, I talk about this more, but I couldn't fit it in. DNA from living individuals can definitely give us a histories of their populations. But there have been a lot of events, historical and evolutionary events, that can obscure that history, right? One example is, unfortunately, the events that took place following colonial, or at the beginning of colonialism, right? Post 1492, there have been a lot of population events, including, population bottlenecks resulting from genocide and colonialist practices that have really deeply impacted Native communities and their genetic compositions. And so if for particular questions, it's great to look at in present day people's genomic compositions. There are some questions that you really need more ancient samples to look at. And this is true globally for all populations, not just in the Americas.
So here's another population Y question, and this is from, I think, Pierre. What are your thoughts on the puzzling fact that population Y signals only found in modern individuals, but not in any individual from early in the middle Holocene analyzed so far, except the Lagoa Santa individual, for whom the evidence of population Y ancestry is pretty weak?
That is an amazing question. I was gonna say Lagoa Santa. We know there's at least one individual who seems to have that population Y. I think I don't know. I think that may have something to do with genetic drift, right? But I wouldn't be so sure that there are no ancient individuals who don't have population Y. I'm sorry, no ancient individuals who have population Y ancestry. I think they're just not published yet, right? So, and that's all I can really say about that. But stay tuned. I mean, this is an extremely exciting, evolving area of research, and I think it's really cool. We'll find out. I mean, you guys, this is the best time to really be following this, because things are changing so rapidly. But then again, also please keep in mind, the gaps in our understanding geographically, temporally, they're huge, right? And I want to reiterate, there is no single ancient genome there's no ancient genomes that is worth causing harm to indigenous peoples. So we have to keep in mind that filling the gaps is not necessarily the goal, and it shouldn't be the goal if it comes at the expense of indigenous peoples. So we have to hold these things in balance.
So speaking of filling gaps, this is a question about another discipline, but just in case you happen to know, Michael asks, do you know whether there are any underwater archaeology efforts underway to look for submerged archaeological evidence from Beringia?
Oh, from Beringia, I don't know. I know that there are folks who are doing core sampling, or have done core sampling in Beringia, but that's kind of like needle in a haystack, right? Like that you would find artifacts in particular places. It's great for reconstructing paleo environments, though, and this is how we know a lot about what potentially there are these refugia environments, right? I don't know if there's any underwater archaeology going on in Beringia. I wish there would be. That would be great. I do know archaeologists are looking at layers, geological layers, which date to the LGM that have been brought above the water because of isostatic rebound, which is this whole thing I won't get into. But there are archaeologists looking at those and looking for sites there. So hopefully that will bear fruit. We'll see.
Excellent. Thank you. Here's a question. Is there a genetic link? This is from Dave. Is there a genetic link between the ancient peoples of North America and Europe or Russia? Arctic Circles people, he asks. Arctic Circle peoples.
Do you mean like present day?
Let me make sure I read the question. Is there a genetic link?
Yes, there's a lot of interactions. I didn't even talk about the peopling of the Arctic. I wish I could, because that's something I'm fascinated by and do research in, but only at 30 minutes. But yes, there's a lot of gene flow going on between populations in the past and in the present, circumarctic populations. Mm-hmm.
Another question from Dave, because it's a nice follow-on, a little bit of a pivot though. How long does it take for an isolated genetic group to become its own genetic group? Are Paleo-Siberians separate from ancient Beringians, separate from native North Americans?
Well, it depends on how you define separate, right? I mean, I don't know what, that I can't answer that question. We can tell genetically, we can tell the difference between Paleo-Siberians and ancient Beringians, which is such a confusing name. The ancient Beringians, the population that lived in Alaska, and unsampled population A, and so forth. Like, you can tell the difference genetically between these groups. but they're really similar. They're really closely related. They're like family members, right? And it's only by looking at complete genomes with millions of different loci, right, that you can actually get at these differences because they're so subtle.
So here's another question about different kinds of ancient DNA, if you happen to know anything about this. So Tim said, I would love to hear a bit about ancient DNA recovered from coprolites, both human and other, food, hindgut flora.
Yeah, I'm a big fan of coprolites. Yes, some of the early studies of mitochondrial DNA, which I didn't even have time to get into, were recovered from coprolites at the Paisley cave sites. And I guess there's some people who find that a bit controversial, but they, you know, I haven't been persuaded that they're not accurate. And there's, I think there's a lot of other coprolite research going on. I haven't heard about any human whole genomes being recovered from coprolites yet. Maybe I'm wrong about that, but nothing comes to mind. But certainly it's a really great potential source of DNA, and I think worth exploring for sure.
Nice. So here's a question from Jeff. Contrary to the Beringia model, the genetic and ethnoarchaeological data better supports a multiple migration model. Specifically, the Algonquians and Na-Dene, I'm probably pronouncing that wrong, have different roots in Asia from each other and from other Native Americans. And he asks, why do you not entertain a multiple migration model?
So I don't, okay, first of all, I'm not persuaded by those studies. So I'm just going to put that out on the table. Some geneticists are, I'm not. We'll wait and see. I'm not opposed to a multiple migration model. The question is multiple migrations from what ancestral group and how many and when, right? And when I talk about this ancestral Beringian population, I don't know where it was. I think it was in Beringia, but maybe it was in East Asia. I've got some issues with that, mostly because when populations are near each other, they tend to have sex. And we see this population being pretty definitively isolated, but we don't see evidence of, we see evidence of later gene flow that can change the genetic composition of some of these populations. But I don't think that there was one like massive migration like is posited by the Clovis first model. Instead, I think about it more as small groups moving in to the population, extended families, possibly from multiple refugia, which could have given us this pattern that we're seeing where we see all these population lineages diverging during the LGM. And it may be that these multiple small groups are moving in and then sometimes moving back, right, as people do, and they're moving seasonally. Migration is a messy process. It's not, as I say in my book, you don't just draw arrows on maps, and that doesn't encompass the whole dynamic process of migration. People are moving around, encountering each other, going back. And so I think it's an oversimplification to say we have these discrete migrations of these discrete groups, or even we have this one massive migration. I don't think either is an accurate depiction for what really happened. Oh, Brianna, you froze.
So here's a question that might follow on from that. And so, oh, can you hear me? Can you hear me now?
Yes, now I can. Yep, you're back.
Okay. Sorry about that. So I was saying this is a question that kind of follows on from that, thinking about like, how do you think about genetic separation? So Jamie asks, could the genetic separation happen at the moment they were no longer commingling, not just because one group walked away from another group?
Yeah. I mean, the way I kind of think about what was going on in Beringia or wherever they were during the LGM, and maybe they weren't in Beringia during the LGM, right? We have to reconsider all of this, but let's go back to this working model. I mean, I think that people were dispersed over a broad geographic area. And, you know, if you think about it, you've got different regions that probably had, you know, different climates, and people are moving between these refugia, these regions, right, to exchange marriage partners, to trade, just living their lives, right? That's why I talk about, if people were in Beringia, it's not a bridge, it's a homeland, twice the size of Texas. And so people are dispersed across this wide geographic space, and that actually does have impact on genetic populations, right? You're going to see this genetic structure emerge from limited gene flow, restricted gene flow between these different groups. Now, I'm waving my hands a whole lot because we have a lot of work to do to actually figure out if this is what was going on. But that is a pretty good working model to test.
So here's a good, I think, high-level methods question. So Kelly asks, given indigenous migration and displacement pre-colonially, how is ancestry of remains determined?
That's a great question. So there are genetic variants that are present only in the first peoples of the Americas. And this gets at a really big question about identity and ancestry and genetic variation. So I really want to emphasize that when we talk about these topics, we're not saying that only people with ancestry from these first peoples are Native American. It is absolutely not the role of any geneticist, including myself, to say who is and who is not Native American, because it's not determined by genetics at all. But when we're talking about genetic histories, the first peoples, the ones pre-contact, right, have genetic variants that we don't see anywhere else in the world. And so you can model population histories on the basis of that. And it gets very tricky, right? Because then you're saying, well, were there Europeans present? Some people say this. It's not, there is no evidence for gene flow from any external population right now, including upper Paleolithic European populations in these models, right? You can actually explicitly test that and using tools of population genetics, and it is ruled out. A lot of changes, a lot of population dynamic changes happen as the result of colonialism, and that is also can be modeled, and it's an emerging area of research as well. And Brianna's frozen again. Oh no. I think she's in France right now, so I don't know what her internet connection is like. Let me see. I'm going to look at the Q&A and see if there's anything else that I can answer while we're waiting for her. Let's see.
I can turn off my video. Can you hear me?
Oh, I can hear you now. Yay.
Okay. I'm going to turn off my video just because my connection's a little stable, and hopefully this will be this will work a little bit better. So I have a question from Sophie. This is interesting, thinking about again, sort of about methods and different materials. Is it possible that there were additional species other than dogs that the people brought with them that might give additional information, other animals or seeds?
Yeah, that is a great question. There were no, as far as I know, no other domesticated animals brought from Siberia, because at the time the first peoples came, they were the dogs were the only animals that were domesticated at that point. But one really interesting area of research is looking at pathogens, because we all carry microbes in our guts, and they may be dangerous for us or they may not, right? But the same thing is true from the first peoples. And so there is a really interesting, again, area of emerging research looking at the pathogens or the gut microbiomes that may have been present, and not just gut, but other microbiomes that may have been present in these first peoples. And can we reconstruct anything about history using those microorganisms as a separate a source of data, an independent source of data? And that is a very exciting area. I did not write very much about it in my book, but I think it's definitely worth looking into. I'll see if I can drop a few papers into the Google Drive on that subject.
Okay, great.
I think we might be.
I'm going to ask 2 more questions, and because we're having a little bit of technological difficulties, but hopefully we can last. Can you hear me? Am I coming through?
Now you are.
Okay, we're going to more questions. I'm so sorry about the technical difficulties. So we'll ask a question. I'll ask a question from Michael. Can you talk about practical impacts for, say, Indigenous health that might come from a better understanding of Indigenous genetic histories?
Yes, kind of. This is not my area of research. I don't do health related research at all, but it is true that understanding genetic variation of and genetic ancestry can be used to inform understandings of the evolution of particular genetic disorders. And there are a number of people who are doing this work. And I want to say that I want to give a shout out to my colleague, Professor Keolu Fox, who is an Indigenous Hawaiian who does this kind of research and advocates for tribes to be doing this kind of research themselves and building capacity among tribes for doing this kind of work, because it can be quite difficult for outsiders to be able to come into a community and do this work without understanding all the nuances of how to do it in an ethical way. And this is, in fact, one of the major pieces of history that's really been harmful, is a history of doing this kind of work by outsiders who have no understanding or no regard for potential stigmatizing research or how they present their work, or no accountability to tribes about the results and how they should be presented. And so this is, I can't really answer your question because that's not really my area of research, but I do think that it is important, but it's equally important that tribes have oversight of this research themselves.
Thank you. I think that's a really fair answer. I'm going to ask one last question, and thank you, Amanda, for jumping on in case my audio is just too distracting and not working very well. So the last question is from Rob, who says, given ongoing and fierce discourse about what it means to claim Indigenous or Native American identity, for example, Liz Warren, Sacheen Littlefeather, federally unrecognized tribes, can you speak to the role of genetics being used or misused by scientists, advocates, activists, or others?
Yes, I'm glad you brought that question up. It's a complex topic. As I said earlier, it is not the role of geneticists to define who is or is not Native American. There is no, as far as I'm aware, and I'm pretty sure this is true, if there are any Indigenous people in the participants, they can maybe jump in on the Q&A chat and correct me, but I don't believe there are any tribes that use DNA ancestry testing as a criteria for membership. Membership in tribes is the purview of tribal governments, and that is definitely not an area where non-natives should have any say. A lot of people have family traditions and family histories about having Native ancestors, and they may use genetics as a tool for testing the truth of these, but I'm very uncomfortable with that. I think that it privileges DNA as a tool for defining indigeneity, and I think I want to encourage everybody who's interested in this topic to read the work by Kim Tallbear, she does a lot of really great critical work in this. And there are a lot of other Native scholars who do that as well. She's a great place to start. I am not Native, and so I am very uncomfortable talking about these subjects. I want to stay in my lane, but I will say that ancestry testing, as I understand it, ancestry testing can be used as a tool to begin reconnection, but it's just a start. And I think that there's a lot of hard work to be done in building cultural ties and legal ties. And again, something that geneticists should not, non-native geneticists should not be speaking to.
All right, thank you. I'm actually going to hand over the closing to Amanda because of my audio difficulties, but I just want to say thank you so much, Jennifer, and I will hand over the closing to Amanda.
Thanks, Brianna. Brianna's been tuning in from France today, so I'm glad we made it almost to the end. I'll just jump in and thank you to our speaker, Jennifer Raff. That concludes today's virtual event, and this season of Hot Topics will resume again in February 2023. So audience, please join me in thanking Jennifer, as well as everyone who made today's program possible. Thanks to our behind the scenes question answerer, Ryan McCrae from George Washington, to our donors, volunteers, all of you viewers, and to our partners who help us reach, educate, and empower millions of people around the world. Thank you, thank you. Check back on our website for more info. I did drop a link in the Q&A where you can find information about our upcoming programs, as well as sign up for the museum's weekly e-newsletter if you haven't already. That's the best way to stay informed on upcoming programs and learn about the museum's research and exhibitions. As well, when this webinar ends, you'll see a survey pop up asking for some feedback about today's program. Please take a moment to respond. We love to read all of your responses. We're super curious to know what topics you might be interested in seeing for future programs, and we appreciate your input. Again, thank you to our participants, and to you, Dr. Jennifer Raff. See you all soon. Bye.